Hello friends, welcome back to another episode on TransConnect. In this episode, we'll be talking about Lewis, MNS, P and Kell blood group systems. So we'll begin with the Lewis system. Lewis system is so special that it is not intrinsic to the RBCs. It is present on the type 1 glycosphingolipids that are then adsorbed on the RBC membrane from the plasma. It was initially discovered in the year 1946. The Lewis antigens, that is Lewis A and Lewis B, is produced by the interaction of two fugosyl transferases encoded by two independent genes, the LE and the SE gene. The secretor status is also associated with the Lewis antigen synthesis. Lewis A individuals are non-secretors and Lewis B are secretors. If we see the prevalence, Lewis B is the more common one. Coming to the genetics, it is synthesized from chromosome 19, the LE gene that encodes for fugosyl transferase 3 or FUT3. The synthesis of Lewis A and Lewis B proceeds from the type 1 precursor chain, that is the chain responsible for secretions. The Lewis A is a monofugosylated antigen while B is a difugosylated which requires two enzymes, FUT2 and FUT3. So if we see the synthesis in detail, I've compared it with the A and B antigen synthesis. In the center, there is the type 1 chain that is beta 1,3 linkage. Now, this chain is responsible for producing the secretors of ABO blood group. So, when there is secretor allele, that is a capital SE, there will be action of fugosyl transferase 2 because of SE and fugosyl transferase 3 because of LE. The fugosyl transferase 2 adds fucose to the terminal galactose, while fugosyl transferase 3 adds fucose to glucosamine, N-acetyl glucosamine. So, when secretor gene is present, both SE and LE acts upon adding two fugose, thereby producing Lewis B antigen. But when only one glucose, that is when only LE allele is present, the fugose will be added to the N-acetyl glucosamine, giving us Lewis A. So Lewis A individuals are non-secretors of ABO, while Lewis B are secretors. The Lewis A and Lewis B antigens are not expressed in the cord cells. The red cell of an infant who has to become a Lewis B adult will initially be Lewis A negative and B negative, followed by A positive, B negative, and finally A negative and B positive. So the Lewis antigens are diminished in the mother during pregnancy. These antigens are found not only in RBC but also platelets, lymphocytes, and other tissues and organs such as pancreas, stomach, intestine, muscle, renal cortex, etc. And therefore, these antigens play an important role in organ transplantation. Soluble antigens of Lewis are also found in the saliva as glycoproteins. These antigens are resistant to fissin, peptide, DTT, and glycine acid EDTA, and therefore their reactivity is enhanced when we do it with enzyme-treated RBCs. Coming to Lewis antibodies, they are naturally occurring IgM. They do not cause hemolytic disease of fetus and newborn because IgM does not cross the placenta, only IgG does. They can be neutralized by antigens in the plasma or commercially available antigens also. The agglutinates, these antibodies agglutinate saline suspended RBCs, therefore they're fragile and easily dispersed. The agglutinated red cells provide a kind of stringy appearance. They bind to the complement and thereby causes in vitro hemolysis also. Hemolysis generally in Lewis antibodies is rare because Lewis antigens that are present in the transfused RBCs will shed within a few days, and Lewis antigens in the plasma transfused will be neutralized by the recipient's antibodies. So anti-Lewis A is rarely detected in room temperature, therefore rare hemolytic transfusion reaction is possible. Lewis B individuals do not make anti-Lewis A because the unconverted Lewis A will be also present in the plasma. The anti-Lewis B is less common and less strong. It is an IgM agglutinin. There are two varieties of examples. One is the anti-Lewis BH, that is, it will react to both Lewis B and H antigens, kind of compound antigen like. And there is another entity called anti-Lewis BL, that is, irrespective of the ABO status, this antibody will act. Coming to disease association, the Lewis B and H antigens are associated as receptors of H. pylori infection and therefore implicated in gastritis, peptic ulcer disease, gastric carcinoma, and mucosa-associated lymphoma. The Lewis A negative B negative phenotype increases the individual's susceptibility to infection by Candida and uropathogenic E. coli. Coming to the MNS system, it was initially discovered in 1927, followed by 47, 51, and 53. This MNS group is mostly low prevalence. Antibodies of low and high prevalence antigens are not commonly encountered. If we come to the prevalence, the phenotype M plus and N plus is more common. Coming to the genetics, the chromosome 4 is responsible for producing the genes GYPA gene and GYPB gene. The GYPA gene encodes M and N antigen and GYPB encodes S and S. The GYP stands for glycophorin. These genes are codominant genes and are homologous, that is, they are almost similar genes. Misalignment during meiosis is responsible for the variants in this gene. Complex variants can also happen. So coming to this picture, this denotes glycophorin A and glycophorin B. Glycophorin A is a 131 amino acid protein and glycophorin B is a 72 amino acid protein. Both are transmembrane proteins. So M and N antigen is found on the glycoprotein called glycophorin A. It is a sialic acid rich glycoprotein. Sialic acid is responsible for the negative charge of the RBC membrane. Now this protein contains 131 amino acids. They are antithetical. They differ in particular positions of amino acid, that is 1 and 5. If the position 1 contains serine, it is M antigen, and its leucine, it is N antigen. If the position 5 contains glycine, it is M, and glutamic acid, it is N antigen. These antigens are very well developed at birth. Since the antigens are present at the outer end of the GPA, that is glycophorin A, it is destroyed by the enzymes, but not affected by DTT alone, AET, chymotrypsin, chloroquine, and glycine acid EDTA. The neuraminidase enzyme also cleaves sialic acid and thereby abolishing the antigens. Coming to S and small s antigens, they are found in the glycophorin B, which is the 72 amino acid glycoprotein. Here, S and S differs in position 29. It is capital S if it is methionine and small s if it is threonine. It is well developed at birth, degraded by the enzymes, same as M and N. Coming to anti-M, it is a naturally occurring saline agglutinin. Both IgM and IgG component is present. They do not bind to complement and do not react with enzyme-treated RBCs. They exhibit dosage phenomena. That is, in a homozygous state, they are more stronger than the heterozygous state. The reactivity of anti-M can be enhanced by increasing the serum cell ratio, increasing the incubation time, decreasing the temperature, and adding potentiators such as albumin, Lis, and PEG. Some of the anti-M examples are pH dependent, that is 6.5. Some react in glucose solutions. So anti-M that are not reacting at 37 degrees centigrade are of nil significance, and antibodies that react at 37 degrees centigrade are important. So units that are compatible at 37 degree and at AHG phase is important. They cause rare HDR and HDFN. Coming to anti-N, anti-N are almost similar to the examples of anti-M. The one thing that differs is the dialysis associated anti-N. So what happens is formaldehyde treated RBCs due to dialysis produce an antibody similar to anti-N. Now the titer of this anti-N will fall once the dialysis has been stopped and the exposure to formaldehyde is over. Coming to anti-capital S and anti-small s, both are IgGs reacting at 37 degrees centigrade and the AHG phase. If the anti-capital S and small s are suspected, what we can do is we can incubate at room temperature and do the immediate AHG without incubation. So these both also exhibit dosage effect but not as dramatic as anti-M. These both antibodies bind to complement and thereby causing severe HTR and HDFN. So antigen negative and cross-match compatible units is important. Coming to new phenotype, this is at the glycophorin B, 33 to 39th amino acid. It is present in the RBCs of all individuals except those who lack the glycophorin B. Now this is resistant to the enzyme. The U antigen is always present when capital S and small s are inherited. So S negative and U negative usually happens when the coding region of glycophorin B is not uh, produced. Coming to anti-U, it is produced in response to pregnancy and transfusion. It is an IgG, thereby causing severe HTR and HDFN. It reacts with U negative RBCs weakly by adsorption and elution. Coming to ENA negative phenotype, this happens when almost all entire or some most of the parts of glycophorin A is missing. There are three types of antibody produced: anti-ENA TS, FS, and FR, standing for trypsin sensitive, fissin sensitive, and fissin resistant. This anti-ENA is notorious for causing HDR and HDFN. Coming to MK phenotype, it is a rare silent gene, near complete absence of both glycophorin A and B. So the individuals will be MN, capital S, small s, U and ENA negative. Everything will be negative. They have reduced sialic acid content, but yet provide a normal hematological picture. Now this MK phenotype is the null phenotype of the MNS system. Coming to disease association and other antibodies, there are other antibodies in the MNS system. So if the antibody is against low prevalence antigens, what we do is we transfuse cross-match compatible units at 37° and AHD phase. If it is an antibody against high prevalence antigens, then we have to ask for a antigen negative unit at a reference labs. Coming to disease association, the MNS system is associated to be receptors for complement bacteria and viruses. The glycophorin A is responsible for pyelonephritic E. coli and Plasmodium falciparum. Coming to the P1, PK and globoside system and related antigens. So initially they were a group of antigens, but after discovering the biosynthesis pathway, they were reclassified into P1, PK system, globoside system and the globoside collection. It was initially discovered in 1927, followed by 1951 and 1959. Coming to the prevalence, the P1 phenotype is the most common one. Regarding the genetics, as we can see here, the entire system is produced starting from the ceramide. The ceramide is converted to beta glucosyl ceramide, followed by lactosyl ceramide. At the lactosyl ceramide, the pathway divides into two separate pathways. On the right, we can see the synthesis of PK, GB3, and a mutated variant called NOR. On the left side, we can see the P1 antigen. If we can closely observe, the P1 antigen and the H antigen has a common precursor, paragloboside. Coming to the phenotypes, P1 and P2, the classic P system has two main phenotypes. The P2 individuals will make an anti-P1. PK is a rare autosomal recessive phenotype. PK can occur either on P1 or P2 background, giving rise to PK1 or PK2 phenotype. The P null is the true globo-null phenotype. The red cells and other tissue will lack P1 and all the globo-GSL, nor is a mutated variety. Coming to the antigens features and properties, they are synthesized by sequential action of glycosyl transferases. The P1 and ABH antigens have a common precursor. The P1, P and PK are seen in RBCs, lymphocytes, granulocytes and monocytes. The capital P is also seen in platelets, fibroblasts and epithelial cells. They are not identified as secretors. They are enzyme resistant. The P1 antigen is poorly expressed at birth. The strength varies among individuals. Homozygous, the strength of the antigen is stronger, and in blacks also the strength of the antigen is stronger. P1 deteriorates on storage. Therefore, if we type an old RBC, it will give us false negative. P1 and PK is also seen in hydatid cyst, which is an interesting point, and it is also seen in bird liver fluke disease. Soluble P1 are commercially available to neutralize anti-PN. Anti-PN is a naturally occurring IgM. Since it's an IgM, we don't have HDFN. It is seen in P1 negative individuals. It is a weak, cold reactive saline agglutinin. But there are some strong examples that react at room temperature also, and those react at 37° centigrade also. Anti-P1 is generally seen where there is an inconclusive antibody identification. But if we cross-match and give compatible units at 37° and AHG phase, it will not be an issue. Coming to anti-PP1 PK, it was originally called anti-T PJA. It is produced by small p individuals and it reacts with all phenotypes except small p. It can be split into three components as an anti-P, anti-P1 and anti-PK. It is a mixture of IgG and IgGm, has a wide thermal range, binds to complement and causes severe HTR and HDFN. It is also associated with spontaneous abortions in early pregnancy. Coming to an allo antibody anti-P, it is a component of the anti-PP1-PK. It is a naturally occurring antibody, potent hemolysin with a wide thermal range, therefore very significant clinically. There is another entity called autoantibody anti-P, which is associated with peroxisomal cold hemoglobinuria. The anti-P specificity is associated with a cold reactive IgG autoantibody. It is a biphasic hemolysin. It will bind with a complement at cold temperature and lyse the RBC at warm. This can be demonstrated by Donath-Landsteiner test. Coming to Luke antigen and antibody, it has a 98% frequency, very common antigen. It is expressed in cord cells, resistant to chemical and enzyme treatment. Therefore, its expression will be enhanced. Antibodies seldom occur because almost everybody will have this antigen. Anti-LKA is an IgM reacting, which will react at room temperature or below. There is no reported transfusion reaction of HDFN. Coming to PX2 antigen and antibody, it is present in more than 99.9% on all cells. It is present on cord cells. Enzyme treatment enhances expression. Antibodies seldom occur, but it is a naturally occurring IgG. There is no significant HDR or HDFN. Disease association, anti-P1 is associated with parasitic infections. Anti-PP1-PK is associated with early abortion. An autoantibody against P is seen in PCH. The P antigens act as receptors for uropathogenic strains of E. coli, Shiga toxin, E. coli associated hemolytic uremic syndrome, and human parvovirus. Coming to the Kell system and the KX system. So it has totally 36 high prevalence and low prevalence antigens. The anti-K was first discovered in 1946. The null phenotype K0 helped to expand the discovery of other antigens. If we observe the prevalence, the small k is more prevalent. KPA and JSA are low prevalent antigens, and KPB and JSB are high prevalent. Coming to the genetics, it is synthesis from chromosome 7. Single base mutations is responsible for different antigens, while point or frame shift or splice site mutation is responsible for the null phenotype. There is an interesting rule that occurs here. Only one haplotype will code for one low prevalence antigen. If we can see here, KPA and JSA are both low prevalence, but they will not exist in the same haplotype. The Kell glycoprotein that is synthesized is a 731 amino acid protein, such a large protein, but there is only one span of membrane. The N is intracellular and the C domain is extracellular. It belongs to the neprilysin family of zinc endopeptidases. There is another protein, there is a covalent bond with an XK disulfide protein, which is a 440 amino acid, but 10 spanning. Kell expression depends on the XK protein, and when this protein is absent, it causes something called myeloid syndrome. We'll see in detail. As we can see here, this is a structure of Kell and KX. If we can see, the Kell protein has more than 700 amino acids, but only one span, because most of them form a big cloud outside the cell with multiple disulfide bridges, but there is only one single disulfide bond that connects the XK protein with the Kell. The XK protein has 400 amino acids, which is less than KL, but spans the membrane 10 times. In the XK protein, both the C terminal and N terminal are intracellular, while in the Kell protein, the N terminal is intracellular and the C terminal is extracellular. The antigens are present only on RBCs. The associated XK protein is present in other erythroid tissues, brain, lymphoid organs, heart, skeletal muscle, etc. In the fetal RBCs, capital A is detected at 10 weeks and small k is detected at 7 weeks. They are well developed at birth. They are not denatured by fissin and peptide, but destroyed by trypsin and chymotrypsin. They are also destroyed by other thiol-reducing agents and glycine acid EDTA. The capital K is next to D in immunogenicity Anti-K is produced by pregnancy and transfusion. Capital K has a low prevalence. So there is less chance of a person to receive a K plus unit and develop antibodies. And compatible units can be found even if anti-K develops. Small k has high prevalence. That is, antibodies seldom occur. K negative individuals are very less, and the likelihood of them getting a transfusion and getting immunization is even less. So coming to KPA, KPB and KPC. KPB is a high prevalence antigen. Mutation of this causes KPA and KPC. Coming to JSA and JSB, JSB is a high prevalent antigen while JSA is a low prevalent one. Anti-capital K is seen commonly after ABO and RH antibodies. It is an IgG which is reactive at AHG phase and is produced with response to transfusion and pregnancy. There are IgM examples that occurs with E. coli infection. The most reliable method is to detect is to do the indirect antiglobulin test. Potentiators such as PEG will increase the reactivity. It is implicated in severe hemolytic transfusion reaction and it is associated with severe HDFN also. Antibodies to KPA, JSA and other low prevalent antigens, it is rare because it is low prevalent and only a few people are exposed. It is usually detected when there is an unexpected incompatible cross-match and in cases of HDFN. The characters are similar to anti-K. Coming to antibodies to high prevalent antigens like small k, KPB and JSB, they are also rare because only a few people lack the antigen. Detection is easy, but finding an antigen negative blood is difficult. So we have to encourage donors such as siblings or look for rare donor inventories. Coming to K0 phenotype and anti-KU. K0, as the name suggests, lacks all the Kell antigens. There is an antibody called anti-KU. U stands for universal. That is, it recognizes all the Kell antigens on RBCs except K0. It causes hemolytic transfusion reaction and HDFN. We can artificially synthesize K0 by treating the normal RBCs with DTT, AET or glycine acid EDTA. Coming to the KX system, there is only one antigen KX. It is present in the XK protein. They are present on all RBCs except the myeloid phenotype. The K0 and the K mod phenotypes have high levels of KX. The Kell antigens, when they are denatured, the KX expression increases. Coming to myeloid phenotype, absence of the XK locus. The phenotype is generally K null, but there is weak expression of small k, KPB and JSB. They can be detected by adsorption and elution methods. This is an X-linked phenotype. The RBCs lack KX and KM with marked depression of other kill antigens. There is weakened expression of all the antigens. The features are acantholytic RBCs, that is irregular shaped RBCs with protrusions, and there is a decreased deformability and reduced in vivo survival. There is a chronic well-compensated hemolytic anemia, that is reticulocytosis, hyperbilirubinemia, splenomegaly, and low serum haptoglobin. In the Meckelard syndrome, there is serological and hematological features of the phenotype, along with muscle and nerve disorders. Therefore, this is called as the neuroacanthocytosis syndrome. Slowly progressive muscular dystrophy will be present beginning at the age of 40 to 50 years, leading to cardiomyopathy and cardiomegaly. There is areflexia, that is loss of deep tendon reflexes to choreiform movements. There is elevated level of creatine kinase MM levels. The Meckelard phenotype can be classified into two groups, one with chronic granulomatous disease and one without chronic granulomatous disease. So Meckelard individuals with CGD disease will have an antibody called anti-KX plus KM, while without chronic granulomatous disease will have anti-KM. If we observe the reaction, take a K0 cell. The K0 cell will show a strong reaction with anti-KL, that is with chronic granulomatous disease, but it will not show a reaction with anti-KM. Altered expression of kill antigens can occur in the following situations. Well, first thing is Meckelard phenotype, we saw in detail. People who are Gebrich negative phenotypes. Gebrich is another blood group system. Those who are Gebrich negative will also have a weaker expression of kill. People with autoimmune hemolytic anemia. When the antibody is directed against kill, there will be reduced expression. Antibody strength will return to normal once the anemia is resolved. This phenomenon is very common with Kell antigens. Streptococcus faecium infection, K negative individuals will acquire a K-like antigen, expressing weak antigen expression. So K mod, that is phenotypes with weak expression of Kell warranting absorption and elution studies all come under K mod. So reduced Kell is always associated with excessive Kx expression. Thank you so much. We'll meet you in another episode.